to them. Okay, so you can see that my topic or my interest has been the sustainable. That remember that yesterday we were talking about sustainability, which is a big key word these days, and that's so my area has been the sustainable, eco-friendly approach of in-plant green engineering of gold nanoparticles. And you see there are a lot of buzzwords in there. And that one of the key thing is that you need to use buzzword in order to get it funded from the funding agency. And what I'm talking about that it has been funded through National Science Foundation, had for a 3 years grant that expired, and then currently I have another 3 years of NSF, second NSF grant, basically in the same area. Okay, see that many postdocs, Dr. Nilesh Sharma, Ajay Jain. Nilesh, he finished the postdoc. Currently, he's an instructor in my previous university in Western Kentucky University. And then Ajay finished his postdoc, came to India as a Rama Lingam Fellow, work in ICAR, no, not ICAR, IRI in Pusa Institute in New Delhi, and currently he is a deputy director of biotechnology, I believe, in Amity University in Jaipur. B. Sinilal, Bhaskaran Sinilal, he is from Kerala, and currently he is assistant professor in one of the state-supported institution. Sneha Krishnamurthy, Sneha did her PhD from South Korea and came as a postdoc in the lab, and currently she is faculty in University of Tampa in Florida in USA. Devesh Shukla, he had PhD from NBRI, National Botanical Research Institute in Lucknow, and then he finished and came back here, and I believe he is still looking for a permanent job, but he is also consultant, doing some consulting job with certain companies and so on. And there are, you see the list here, these are the partial list. I had many undergraduate and graduate students working on the project. Some of those finished their PhD, working as a faculty. Some are still in PhD programs. And this guy, Adam Ismail, he was undergraduate and he did research in my lab, a great guy with the TEM, transmission electron microscopy. Did several initial work the lab, and currently he's the pilot. So he flies the plane around for Delta Airlines. So it's not only you go to the academic line, you can go anywhere you want to. Okay, so I'll talk about introduction, brief introduction. We have been talking for a couple of days, and then we'll talk about the plant's ability to synthesize nanoparticles, the different ways you can synthesize these nanoparticles, conventional ways, and also that using the plants, and also the ability to control the shape and size of those nanoparticles. And plants can do it, that just by expressing certain genes, that stopping them or expressing them, you can make whatever you want to in terms of shape and size of these nanoparticles. And we'll talk about the different ways we have analyzed certain genes, and we try to characterize them, and then eventually we'll talk about a conclusion, or just before the conclusion we'll also talk about how do we isolate these nanoparticles, because it is being synthesized by the plant. You've got to have a great method by isolating them, then you can study their applicability, that what they can do for any ideas we have talked extensively yesterday. Okay, so just by just the definition, the creation of functional devices in the nanometer range. We talked in a lot more detail, so I'm not going to go in great detail, but briefly talked about that. You remember seeing this slide earlier. Of course, you know, if you look at here, that where is the nanometer over here somewhere, that you're talking about 1 billionth, 10 to the power -9. And then if you go further down here is picometer. You see the, just to get an idea that, you know, what size we're talking about, you know, that your DNA structure is about 2 to 3 nanometer in size and so on. There are, can you guys see from the back? Okay. There are many applications. We talked about that yesterday, that, you know, that we talked about energy, biomedicine, cosmetics, and so on. And basically they are listed here, that, you know, drug delivery system for these nano materials, anti-corrosion barrier coatings, UV protection gels in cosmetics. We talked about that. Lubricants, you know, that being used. New fire retardant materials that, you know, that they can minimize the fire problem in any kind of industries and any kind of household. You're talking about new scratch, abrasion resistant materials, and we talked about the paint, you know, remember that Mercedes car, we talked about that yesterday, and super strength fiber and film. So we have already talked about that in a lot more detail. There are some, you know, basically taking those, putting into a small slide over here, that, you know, you see here that in automotive industries, different kind of application, chemical industries, engineering, electronic industry, construction, many applications. In textile, we talked about that. In fabrics, energy, we talked about that. We talked about cosmetics, food and drinks. You remember that, you know, that donuts having titanium dioxide. You know, that we talked about ice cream, if you remember that, with a lower calorie. We talked about household items here. Sports, we talked about, you know, that tennis racket, tennis ball, and so on. Yesterday we talked about that. So there are many applications. Some here that, you know, that when we started this project, you know, that, you know, we were thinking about biomedical applications, that how can we synthesize these nanoparticles in the plant by eco-friendly method and use them for biomedicine here. So if you look at some of these, the medical prosthesis, reduce stiffness mismatch, prevent fracture, tissue engineering, you know, you need biocompatible with the native tissue structure, that, you know, whatever you use them, it can be absorbed by the body. It can be biodegradable, so it doesn't become, because, you know, remember that the key word earlier in the title we talked about eco-friendly here. So you want them to be biodegradable, not producing them and then you create tons of these product and then that becomes a problem, what to do with those, you know, the compound came out from those the reactions and so on here. Wound dressing, biodegradable, polymeric fiber spray. You know, as we talked about that, you know, these days many incision that they are using different kind of nanomaterials, glue structure. So basically they will make the incision and then they will not use stitches. In fact, they will just put the glue. So, you know, minimize the scar development in the future and so on. Aids the formation of normal skin growth. Of course, it does it. Prevent the formation of scar. And then we create a non-woven nanofibrous membrane with a pore size between 500 to 1,000 nanometer. In nanomedicine, as you synthesize them, you're looking for potential application in medicine. We talked about drug delivery. We had a long talk about that yesterday. Medical imaging, that being nanotechnology being used in developing different kind of microscopy, medical imaging to detect the diseases as early they can in order to treat them. Diagnosis and sensing, therapy, and so on. So all these, we talked about that. So all these nanoparticles, they are doing good things. At the same time, they are also doing the bad things, because one of the problem is that using these nanoparticles, they aggregate. So you start with the small, but time to time they will aggregate and they will become big. And once they become big, then that's where the problem is. And then of course, that we talked about that, and we also will be talking in the future that their potential toxic effect. That how can we make sure that what is the toxic level and so on, and develop those kind of rules and regulation for further use. Food, if you remember that, this one we talked about that, that using the nano-scaled emulsion, and of course the fat content in this ice cream is reduced from 16% to 1%. So it is good, but then if you are taking them, what they are going to do in the future and so on to our body is we still have to know about that. And of course, this technology being used for several other kind of product, mayonnaise and so on. This one, did I talk about? No, I don't think I talked about that yesterday. That remember that encapsulation of additives, and if you remember that, you may recall these omega-3 from the fish oil, because omega-3, that it is a supplement. A lot of people take it, and this is good for your heart. Okay, so and it also gives you some sort of odor. So people, when you take those tablets, you feel it, that taste and odor in your mouth. So what they have done is they have taken the oil and put them in the nano capsules to prevent them from oxidation and remove any kind of fishy odor. Okay, and it is essentially, they have made this bread here having the omega-3 in there. Again, using nanotechnology here. These are the methods that that we use them to synthesize these nanoparticles for any kind of potential application. And if you look at here, that the conventional method, the physical method, chemical method, that most of the nanoparticles you buy from any vendor, mostly these coming from the chemical method here. And then there are some green synthesis using bacteria, using plant extract, and this is the one I'm using here in the plant synthesis here. And we'll talk about that here in a little bit more detail here. If you look at the extract, that you take the plant and you crush it, grind it, and then you add certain salt, you will get those nanoparticles. But then the problem with this one is that you don't have control over what kind of nanoparticles you are getting, because when you crush it, it's dead, right? But in this case, it is the living system here. So we are synthesizing within the plants, and as I mentioned earlier, that we can control the shape and size, because remember that, that if you are making a metal nanoparticles, think about that. And in this case, we were doing the gold nanoparticles. Many of these metals are toxic to the plant. So if the plants are growing in those toxic chemicals, it is kind of a stress response for the plants. And you'll see later on, essentially here. So basically, that the different kind of stress response will be generated within the plant, and by manipulating those stress response, you can get the different shape and size of these nanoparticles. So this is the one I will be talking in lot more detail here. By using these conventional that you generate a hazardous byproduct, and I'm not sure over here in India, but in US there is a EPA, the Environmental Protection Agency. It's run by the federal government, central government. So if you develop some sort of industry, and part of the industry, if you develop some sort of these hazardous byproduct, then they are going to be asking, okay, so you are generating these tons of these byproduct. What is your goal? What you are going to do with them? Because you need to have some sort of description in your plan that will be doing these, or will be handling these hazardous chemical by these method. Otherwise, somewhere it will be dumped, and they don't want that. Another thing is, in the conventional, that there is a large input of energy is required. You need to use a lot of energy, and then environmental contamination. And another issue is the aggregation, because you'll see that when you synthesize these over here, then you aggregate them, in the clump, all these nanoparticles. Okay, so let's talk about those methods here. That 3 methods we talked about, that the physical method, chemical method, and biological methods here. In the physical, just briefly, not going in detail, but in the physical, you can use the mechanical method, or you can use the vapor method here to synthesize these nanoparticles. In mechanical, high energy ball milling, melt mixing. In vapor, physical vapor deposition, laser ablation, it's quite often being used. Sputter deposition, electric arc deposition, and implantation. If you think about the chemical one, and I just put this one for you guys to from the ACS journal, American Chemical Society publication. Chemical Review came out in 2016 about a nice article about the nanoparticle chemistry. So if you want to know more about that, you are certainly welcome to use them. Okay, so what are the chemical methods? These are the simple techniques. It's like any chemical reaction. That's what it is essentially. Inexpensive instrumentation, low temperature synthesis, doping foreign atoms or ions is possible during synthesis. You can make large quantity of these materials, variety, shape, and sizes, and you can self-assemble, or patterning is possible. You can design how you want to put them together. Okay, and this is, as you can see, this is the chemical method here. It's a reduction method, taking a metal salt, reduce them, and you see, look here, that chloruric acid, and then you basically treat them with the sodium citrate, and then you get that, look here, the gold nanoparticles, but you see you are making all other byproduct here. So you need to think about what to do with those byproduct and so on here. Okay, you can, depending upon the size of those nanoparticle, you can see that that they will give you a different color here. And if you look at here, that if it is small, you get this color. A small size nanoparticle, you get this color. But if you go for the larger, size particle, then you color it changed over here like this here. So you can look the size by using different kind of method, or from this color you can figure it out that general use that what is the size of these nanoparticles. So nanoparticles, gold nanoparticles can be stabilized by repulsive Coulombic interaction. Also stabilized by thiol. So you make these nanoparticles, but you need to stabilize them. So you need to put some sort of coating around that. Okay, and you need to know what kind of coating, because that coating will determine that, where do you want to use them. The coating should be such that, you can, if you want to use it for drug delivery, then coating should have the different functional group by which you can attach the drug molecule in order to direct it to the cell, and so on here. So you put some sort of capping agent on top of that in a smaller manner. Many of these metal nanoparticles, silver, palladium, copper, and other, the iron and so on, zinc and so on, can be synthesized this way here. Also synthesis of semiconductor nanoparticles by colloidal route. That we have wet chemical route using appropriate salts, that sulfide semiconductor like cadmium sulfide, zinc sulfide can be synthesized by co-precipitation. And if you look at here that you want to make it zinc sulfide nanoparticles, you do the reaction. You are treating with the hydrogen sulfide, and then eventually you get the zinc sulfide here. Okay, the last one we talk about is the biological methods, and biological methods are green synthesis method here. That is our interest. That's what we wanted to study, and then you can use this microorganism like that fungus, bacteria, and so on, and lots of articles coming out from India that they are using these microbes. And I do remember that at the beginning of 2000, 2001, 2, 3, a lot of articles came out in good journal, Nature Biotechnology, and several other good journals where that these fungus were used to synthesize, because those were the early days to in the area of nanoscience or nanotechnology, and a lot of good articles came out. And those articles, if you want to publish it now, it's difficult to go to the Nature or any other top-notch journal. Okay. So if you look at here that that using fungus, yeast. Sorry, this is yeast, not yeast. Okay, bacteria, actinomycetes, plant extract. That when you take the plant tissue and extract it, basically that in extract containing different kind of enzymes, and they are the one going to reduce that compound and so on. Microorganisms. These microorganisms being used to make these metal nanoparticles, and you can synthesize these microorganisms. They can produce certain enzymes, secrete outside, and then outside you can see all these nanoparticles will be made. And then you have cell-metal interaction. It's quite complex, because you don't know what kind of enzymes are coming out from there, and what kind of metal particles they will be making. And then later on, once you prepare them, then you can extract them out for further applications and so on. Pseudomonas being used, bacteria being used because they found out that silver mines, because they didn't know that what kind of bacteria to use them, and they said that that Pseudomonas, you can find them silver mines, and that they started using it, hoping that they will be able to make certain silver nanoparticles. So they, yeah, say again. Yeah, natural. Yes, of course. Yes. I mean, there are two ways, that either you can do a lot of experiment and try to use different kind of microbes and see which one is going to work, okay? But that will take a lot more time. So the best one is go to the mine site and try to isolate some of those, because why they are growing over there? Because they have some sort of resistant, or they can metabolize those metals. So it's easy route, that's what it is. So they can, they can use these pseudomonas to make these nanoparticles, silver nanoparticles, and then eventually they got that 200 nanometer size of these nanoparticles here. Low concentration of metal ions like the gold, silver, etc. can be converted to metal nanoparticles by other bacteria, which is called lactobacillus, which you find them in buttermilk. Fungi, fusarium being used. You can use them to make gold or silver nanoparticles. There are some extremophilic actinomyces. They can produce the gold nanoparticles. There are semiconductor nanoparticles like cadmium sulfide, zinc sulfide, lead sulfide, etc. can be produced by many different types of microbes. Sulfate reducing bacteria, they can also be used to synthesize these nanoparticles. So you can see that there are many types here. Desulfobacteriaceae can make 2 to 5 nanometers. See that that some can make a smaller size, some can make a larger size. Okay, Klebsiella can be used to make a cadmium sulfide. Cadmium nitrate can be mixed with the bacteria, and then eventually you can make cadmium sulfide here. Okay, cadmium sulfide nanoparticles, and size range is going to be 5 nanometer to 200 nanometer in size. Okay, so that was about using the microbes. Right now we are using the plant extract as you can find many articles regarding this. And this one is essentially talking about geranium plants. And so they took the geranium here and grind them and used them to synthesize gold nanoparticles. Okay, you can also use certain plant associated with a certain function. There are certain fungi associated with different kind of plants. You can use those fungus to make also different kind of nanoparticles here. Okay. This is the protocol, and I believe you guys did that yesterday in the lab. Look here, that you take the tissue, you crush it, you boil for 1 hour. Boil in the water for 1 minute and then cool it, decant it, add gold salt in the solution, and then you can make these gold nanoparticles pretty soon. Okay? What I am interested is that, so we decided, because our goal, the reason we used living plant system, because our goal was to convert, in certain cases you need only certain size nanoparticles, or you need triangle shape, or you need rectangular shape, or you need circle shape. So we wanted, that when you synthesize, you saw that, that leaf extract, plant extract, are using the bacteria or fungi and so on, that you will get a combination of different kind of nanoparticles. Okay, but we were interested that that we wanted to make that, which is rich in one type or one type of nanoparticles with a certain shape. That was our goal. Okay, so we decided to use that sustainable green synthesis using the plant system here. This one is essentially this one is a 100% green process. Okay, plant is making it. It's no contaminants is generated. Sustainable system, because if you saw that some of the reaction earlier, that you are using the metal salt, and from there you are making the nanoparticles, metal nanoparticles, but then you are creating also hazardous chemical as a part of the chemical reaction. Okay, if you are going to be using, and look here that 58,000 tons per year between 2011 to 20. So you're talking about a lot of these nanoparticles. It means you'll be generating hundreds and thousands of these byproduct. What to do with that? You need to have a plan for that, otherwise the EPA is going to come after you. Okay? The regulation is very strong in the US. Okay? It's if you generate something, you need to have a plan what to do with that. Okay? So that's the reason we decided to use this pathway. This is about my experiment, and you see that this is, I'm talking about around 2000 or so, that we started generating the gold nanoparticle, and it was done by a postdoc in the lab, and we were using Sesbania drummondii. Sesbania drummondii is a plant, and you see the it has nice flower. It's like kind of shrubby. Mostly we found in southern area of U.S. Mostly towards the Gulf line, Gulf Coast, where the Mexico is there. So we use this one to synthesize, and if you look at here, is that this is the the picture is not very good. Maybe color is not very good. But if you look at here, that these are the cell here, cell here, and you see these are the nanoparticles here. These are the nanoparticles. You see inside the cell, you see that there is some structure, although it is not very clear, but these are the gold nanoparticles here. And then these are the TM picture, transmission electron microscopy. And the TM we had, this also has EDX attached. It can tell you that that these, what is the nature of these dots you see here, are these particles, and you can see that it gives you the gold peak here. Okay, you see the carbon is used, so you see in that process, so you see the carbon also peak for there. But these are essentially, these particles are gold nanoparticles here. We were using Sesbania because Sesbania was the plant that we find them for to phytoremediate lead. So we had that in the lab and said, well, let's start using it. Okay. So we also, this is the, you see that this is SEM picture, scanning electron micrograph picture. You see the cell structure here, and you see all these white spots. You see, these are essentially the gold nanoparticles. You can see it, and then if you look at the EDX here, you see that big peak of gold over here. So knowing that these are the gold particles here, and you are growing, taking the plants, and you're growing in potassium autochlorate in the salt. So if you provide the salt, then eventually it will be taken up by the plant, and somehow it being reduced to the gold nanoparticles, which we'll talk later on. Okay? And see, this is the control here. In the control, you don't see any gold here. You see there is a gold peak should be here, but you don't see any peak over here. Okay? if you remember that in my couple of days back, we were talking about that, that nanoscience and nanotechnology is an interdisciplinary department. Remember that. I'm a plant scientist, plant physiology, or plant biotech, or plant molecular biology, whatever you call me. But then, if you want to do certain things, either I should have trained myself, in terms of microscopy, TM or SEM, in order to do that, or you collaborate with somebody, or you have someone, a student in a lab, you train that student to do all these things. But the student can be there for a few years, and once they are done, then they are gone. So you don't have constantly need to keep keep a student, keep training them, or you basically find someone who has expertise and you collaborate them. Okay, so you saw the gold peak, and gold peak, you were giving them potassium autochlorate in the medium to the plant. So it was taking, and you don't know that, the gold peak, you saw that gold peak was AU0, or was it some sort of salt inside? You need to find it out. So there that, I had to go to a chemistry, a good friend of mine, he's at University of Texas, El Paso. He's the chair of chemistry department, a great guy, analytical chemist. So, we were good friends, and I said, Jorge, I found out from his publication that he has access to synchrotron. There are about 5 or 6 synchrotron in the U.S. It's run by Department of Energy, okay? And so he had a grant submitted, and he got that grant funded, and he had to go over there couple of weeks. It was somewhere on the Stanford campus, and he has to do all his samples. So I said, can you see that, that whether it is AU0 or it is some sort of AU salt? He said, yeah, we can do it. And so he went there, and basically he analyzed the data, and he used, by using a synchrotron, he used Jane's, EXAFS. Jane's means X-ray absorption near edge structure. Jane's is extended X-ray absorption fine structure. I have no idea what are they, being a plant. But I needed certain answer, and I cannot become from plant science to a chemistry guy to learn all these things. So he helped me, and that's how we had to go about that. Okay, so in Jane's, what it does, it determines the oxidation state and atomic geometry of a bound metal. And EXFS traces the ligand involved in the metal binding by measuring the distance from the X-ray absorbing atom to the next nearest atom. So here what it is if you look at here, this is, if you look this graph over here, and if you look, this is this different kind of line over here. If this is, let's say, this salt here, you get something like this. If it's the hydroxide, you get something like this. If you have potassium autochlorate, you get something like this. So, he basically ran all these samples. And then he also took our sample, grind it, and he basically ran, that roots treated with the 50 ppm gold salt, 100 ppm, and shoots with the 100 ppm, and he gets pretty similar kind of graph in different examples. And then he tried to match these with which of these lines it matches to, and it matches very well with this one here. You can see that, right? And from there you can say that, look here, that the gold salt we see in the plant sample, it is like AU0 or gold nanoparticles. Okay? Yeah.
How does the particle work?
What is
the salt? Gold salt. In what mode is the?
Okay, so basically you take water, okay, And since we were doing for only short time, we were not doing for long term, so we were not concerned about giving all these nutrients in the solution. So it was hydroponic solution. We took the water and we added different salt concentration, and we grew the plant. Yes. Yeah.
So, and you said that near the root zone, I think you could able to find the zero valent growth,
Mm-hmm.
right?
Yes, in the root as well as in the shoot.
In the,
Mm-hmm.
So what is the mechanism or the reaction that is happening near the root zone?
We'll talk about that. Okay. Okay, so these were the AU0, and from there we confirmed that that these were the gold nanoparticles. And so from there we prepare this table, and if you look at here, these are the samples here. See that the gold salt given into the roots, to the roots at high concentration, and to the shoots here. And from there, you can see that it's approximately more than 80% of the gold salt being converted into AU0. Okay, there were some was gold sulfide there, but majority of those were converted into AU0. Yeah. It was okay. So you are asking that whether that we found the same concentration that what we gave in the liquid solution. Yeah, that's a good idea, because if you are trying to, let's say, in many experiments, if you are trying to clean up certain area, then you try that you started with 100% of this, that whether you have removed all these 100% gone to the plant or not. But that was not our goal. We were just looking that that it goes in there and it has capacity. But that's a good idea, that if you are trying to see that how much being gone in there, whether only 60%, 70%, or all 100%. So trying to close the loop. That's what it is. So you see that more than 80%, 80 to 85% being converted into converted into gold nanoparticle. Okay, and then what we did is that we wanted to take the picture of that and look here the TM. We took the tissue, we crushed it, and you can see all these gold nanoparticles. See all these here. We kind of try to, from this, we try to make it out, the different shape here. See, some were like this, some were like this, different shape and sizes of these. And in this case, in our case, that, the majority, if you look at here, it's not very good, but if you look at here, that majority of the gold nanoparticle were between 7 nanometer to maybe somewhere around 25 nanometer. That was the size of the gold nanoparticles. This is a clear picture, basically showing these high magnification of these gold nanoparticles, and it's showing that that EDX gives you a gold peak over here. You see all these other are some of these are, remember that if you recall that plant contains plant matrix contain different kind of chemicals, so you'll find all those, but gold is not part of the plant chemicals. If you apply the gold in the solution, then it will take it up, and then you will see these gold peaks. Another thing is, again, going back to the interdisciplinary department. So another thing we wanted to make sure, because I had seen publication came out from chemistry lab. In fact, it came out from someone, Dr. Paul from IIT, in Kharagpur that he has synthesizing using chemical method to synthesize these nanoparticles, and he was studying their catalytic activity. So I wanted to see that these nanoparticle they can have catalytic activity. So sometime I visited his lab in IIT Kharagpur, and we did the catalytic activity. And if you look at here, this one is the UV visible absorption spectra of 4-nitrophenol in aqueous solution. So that if you have the 4-nitrophenol, you get a peak like this here. But when you add the gold nanoparticles, and in this case, the gold nanoparticle was synthesized by plant. Okay, but this time we wanted to extract them from the plant, but we had a lot of problem. We could not extract them because when you crush it, they were still embedded with the plant matrix. There were plant parts were there. So we decided that how we are going to study it if we cannot find a better method to extract it, and then in that case we thought, why not we crush it very fine, including these gold nanoparticles, and we do this chemical catalytic activity, and of course using the same one. And if you look at here that over time, this is your 4-nitrophenol, over time slowly it degrades and makes it 4-aminophenol. So it shows the catalytic activity, what he has published in his paper using chemically synthesized gold nanoparticle. So we were able to reproduce that thing. And so this is basically 4 or 5 years of work. We were finally able to publish in 2007 in Environmental Science and Technology journal. Okay, so we were this was an early indication for us that plants can synthesize these gold nanoparticles. So we say, well, this is something about only Sesbania, because all these work you saw that was done in Sesbania. So we wanted to see that it is only for the Sesbania or any plant can do it. So we started looking, and you see only few over here, but we looked about 20 or so different kind of plants, and from there we came the top one, and we decided to go with the alpha-alpha. Why alpha-alpha? Because this is something that the farmers can use it. If you wanted to have a long-term application, or you wanted to develop this technology, then of course the farmers could get some benefit out of that. Yeah.
So could you able to find any Hogland solution also? I mean, whether the synthesis is
Yes, right.
cellular or intracellular?
Yes, it's a good point here that he's saying that that when you germinate or a plant, you put them in the solution, in the hydroponic solution, whether you can synthesize. Yet there are some reports where you can synthesize outside as well, and in that case that some of these enzymes might be coming out from the root system and might be reducing the gold nanoparticles. Yes, it could be done. So yeah, okay. So here that that we decided to take alfalfa. We took 15 days seedling. We grow them in hydroponics. We use this technique ICP, inductively coupled photometry here, and then we basically analyze the tissue, see that that whether they can synthesize the gold nanoparticles or not. And this work was given to one of my graduate students, which he published in Environmental Science and Technology in 2010. Okay, and if you look at here, that when plants were given these gold salts in hydroponic solution, and then basically you were analyzing. So as soon as you give them, you will start seeing it. See that 6 hours, 24 hours, then 7 hours. As you take it, let it go for a long time, you can make more and more here. Okay, if you remember that, one day I was talking to you, or maybe earlier I was talking to you, that these nanoparticles, that as based upon their potential role in any kind of application, that that you need different shape, you need the different size here. So we were interested. Okay, so if we start playing with the growth condition of plants, can we change the shape and size here? So we started looking. If you see over here, if you look over here, is see the pH here, pH. You see that the temperature where they were grown. See the for how long? See that 6 to 7 days, light and dark condition. And these were, we analyzed them, and these were the spherical shape, the circular shaped nanoparticle. So majority of the plant makes the circular shape in case of alfalfa. So if you are interested, you say that that hey, I need only circular shaped nanoparticle, then of course you find out which one is the best. And in this case, this is, let's say here at 25 degree. Grow the plant 25 degree because my interest is only circular shape. Majority of those nanoparticles are going to be in circular shape. Okay. Same thing, we started looking for the hexagon nanogold here. And if you look at here that, pH 7.8, we're getting about 16 to 17%. Okay? This is the basic experiment, that initially we did that. We have not done, we wanted to know that where can you find more 10 toward one type of nanoparticle. And then you start playing, start playing with the, game with the plant to try to increase that size. If you look this one here, a rectangular shape. If you look at here that, at 15 degree Celsius, we are making more than 20% of rectangular shape. When we triangle shape, we were making around 3.8, close to 9-10% of the triangle shape. Why triangle? And from this experiment, you will see that later on we decided to focus our shape in triangle. Why triangle? Because I have spoken to you that triangle has relevance in cancer radiation therapy because the way it absorbs light. Okay? So same time when we were doing the same time, a new article came that, where the doctors were using triangle shape and they were having a lot of success. So we said, well, why not we make a triangle here? Okay? And we started doing it. So this is a close picture of TEM. You can see that, that all these different shapes here. Look here. That, TEM is, because they are very tiny, so you can't see them, right? So TEM is, when you do the focus, you can see the shape. That's what we decided, yeah. So you can see that, different shape and size of these nanoparticles. And we were interested in TEM, so you can see that, maybe rod shape over here, so the different kind, from this picture. Okay. So our goal was that, develop a technique by which we can maximize the particle, gold nanoparticle here. So remember that, as I said, that it is, think about that, the gold is not a required element for the plant growth. You know that, right? So if you force the plant to grow in the gold, then it is going to be, it's growing in any kind of metal, and metal is going to be toxic to them. So it is going to generate a stress response to the plant. So if you look at, and this work was done using Arabidopsis. Arabidopsis is model system for the plant. Okay, so if you look at here, Can we, is any way we can make it a little bit darker here? I'm not sure why it is so light. I'm asking you guys. Huh? Hello? Can we make it a little bit darker here? Not sure what. Huh? Can you guys see that? That, can you look here that this is the, shoot system and this is the root system. You can see from the back, right? Okay. Okay, so if you look at here that, that this is Arabidopsis plant growing in full nutrient rich condition. And you can see very nice long root here. Can you see that? Nice, the apical part here, all these leaves and so on. But in this case, this is the nutrient rich and taken out suc minus means sucrose. It needs, certain sugar to get the energy. So the sucrose was removed from there, and you can see compared to the nutrient rich, you see that that the stem is not doing very well, right? And also you see the root is not doing very well. This is in P minus, means phosphorus being removed from the medium. So what do you see here? Phosphorus is one of the essential. Do you remember that? Huh? Yeah. So look here that how does it look here? Okay. In this case, phosphorus and sucrose was removed. See that compared with the control over here. Okay. So we were trying to see that growth here, and in this case, that we were trying to measure the triangle shape, and these were the triangle in nutrient rich in. Sorry about that. In the nutrient rich condition, you see that we are getting about 4 to 5% of triangle here. We are getting about, in this case, we are getting about less than 4. In P minus, in this condition, we were getting about close to 15%. So slowly, remember that earlier I showed you, that was around 8 to 9%. So slowly, we are just by controlling these nutrient, you are certainly turning off and on certain genes within the plants. Okay, and later on you will see it here. Okay, so we felt that that if you remove the P from the nutrient rich, then you can further increase it, and this is essentially giving a nice picture of, showing the nice picture of triangle shape here. Yeah. So basically, when we treated, we took the plant, crush it, and look at into the TEM. Yeah, TEM, and you analyze the data. So several, so So what do you know? If you do the TEM, there are certain rectangular shape there when you watch them. So you basically take several of those from there, and you count each of those, randomly, not just together. Okay, I'm going to count here, count there, and then from there you take the average. Yeah. Yeah, you're right. Yeah, that was came out that because you are looking from the certain angle, and you might see triangle, but it might be rectangular, because the way it is sitting there. So what we did, that the TM has that there are ways by which you can rotate the base to certain degree, and you can look at by rotating that what it is. Yeah. Exactly. Right. So by rotating the base, you can see it from the side what is, because the bottom structure you are not looking, but when you rotate it, you can see it.
Have you used this nano? Have you used the?
Not yet. I'm just giving you a story, and you will see that why did we not use it, and we'll talk about that. Okay, so so our goal was, remember that it's a tedious process if you're taking a plant cell, right? What is the tedious process? You need to take the seed, you need to make sure that it is, you use some sort of mercury chloride or something to clean it up. There is no contamination. You germinate them, then you put them in hydroponic situation, and so it takes a lot of time to do it. But we wanted a way by which we can get these things as soon as possible. So we decided that, and another problem we had in this case is that if you take these, isolate the gold nanoparticle, is that to crush it. Of doing it, and we still have problem because of a lot of biomatrix there. System, plant embedded was there, and we cannot separate it. So we decided that we need to find a better ways by which we can easily remove it, we can purify, and once you purify, then you can go back and study that what kind of relevance in terms of application it has. Okay, so at that time there were a lot of, well, I shouldn't say a lot, about 15 or 20 labs around the world. They were working on this area, and that people, chemists, that analytical chemists, the plant biologists, everybody was trying to find a better way to extract them. Unfortunately, nobody was able to do it, even including our lab. Okay, so a lot of people just dropped it because, you can synthesize, but if you cannot isolate in pure form, then there is use it. We from National Science Foundation, we were a little bit more persistent compared to other groups. We were constantly working, and luckily we were able to find ways to extract them, to purify them, and we have fairly pure stuff now. Okay, and then I'll that some of the data later on. But so we kind of stayed with that, and that is the only. In fact, that publication should come out pretty soon because it has been very minor comments came through, and it has been should be accepted any time now in a scientific report. Okay, so we decided to go through the tissue culture, the cell suspension culture. So this data I'm showing is cell suspension culture, and if you look at here, the different shape, okay, different shape of the nanoparticle, and if you look at here, that if you look this one here, we were interested in triangle shape, so we increased close to 35% here. Okay, where when we treat them with pH at 4.2. We are still working, because our goal is to take them, higher than 80 or some percentage, higher than that, so we can get pure stuff to do any kind of analysis in the future. So we got here in this case, you see that about 35% at pH 4.2. These are some of the TM picture. You can see all these triangle shape over here and so on. So from here, this graph, you can figure it out that, if you are interested in spherical, spherical is the majority you can get. If you are interested in, let's say, hexagon, if you look at here, hexagon is about 5.7. This is hexagon. So, that we can further extend it and so on. Another concern was that, okay, so coming out from a university, coming out from an academic line, yeah, fine, that you make these particles, you can use it, but then for us that we wanted to know the mechanism, how it is going to be made, or how plant does it, because that's what coming out from the university or being a faculty, that's what we try to do it. How does the system work here? So we decided, that what is the mechanism for gold salt to be reduced into the gold nanoparticles? Because remember that, what is the gold is not one of the essential elements. It is not needed by the plant here. So how does it get inside the cell? We still had no idea about that. Does it use some sort of transporter? And if you remember that, in going back and look in the literature, that the arsenic is the major problem, but it is not needed by the plant. But the arsenic gets inside the plant by using the phosphate transporter. Phosphate is needed, so they are there, but then it stops the phosphate and use that transporter to get inside. And that's what causes the toxicity here. So we wanted to know the molecular responses that the plant to when we treat them with the potassium autochlorate here. And we decided to use Arabidopsis. So going to a little bit more detail, and if you look at here, this one is essentially looked at the different ppm concentration of potassium autochlorate, the growth of these Arabidopsis seedling. And if you look at here, 0, 1, 10, and then when you increase the concentration, what do you see here? There is a decrease in the growth rate of the plant here, right? So what do you see here, that when you increase, goes to the 10, then you see that there is an increase in the growth rate. So we found out at the lower concentration there is a hormesis effect here. At the lower concentration, somehow it is increasing the growth of the plant here. And if you remember that, we talked about that, some of the cases that, certain these nanoparticles were inducing the growth. Do you recall that? Okay. Are we dealing with something like this here? I mean, there is a question there. We have not tested, but that at lower concentration there is a push to grow further. Okay, so hormesis here, and if you look at here, that the same root and shoot growth being plotted in this graph over here, and the shoot concentration, and then if you look at here, if you look the further the root system here, 0, 1, 10, you see that increase over here. You see a lot more, roots are coming out, right? But as you increase that, you see that lot less coming up here. So it is becoming a toxic over here. So basically from here that, we say that up to 10 nanometer there is a hormesis effect here. Okay? So we got into a little bit more physiology, essentially, while studying this thing here. Okay? While doing so, the TM, a lot of places they have TM. You're close to, we just got about at my old place a new grant to buy a new TM. You're talking about $450,000. Okay, which is a lot of money here. But some places do not have TM. My new place does not have TM, but there is an institution right beside our institution. They have TM, and I can use over there. So this new technology developed by a physicist at Auburn University, and I found it out. I went there, and they basically analyzed my tissue, and we got a publication out of this. And look here, this is called a hyperspectral microscopy system here. And if you look at here, is all these red dots here. These are the gold over here. These are the gold over here. Okay, so I send them sample, and they did it for free and so on. And we call that that technology as CytoViva. That's what they call it. At the section.
At the ultra microscope, and they took the specimen and they
Yeah, they gave us a protocol how to cut them and how to save them. And so we cut it and we saved them and gave them those samples and they analyzed it. Yeah, so we had to do that. Yeah. Depending upon somewhere 10 to 40, 10 to 40. Okay, so we wanted to know the molecular mechanism, that how does it get inside, and that time we had no idea. We didn't know that, what kind of molecular mechanism helping these potassium salt to get inside the tissue in order to be converted converted to gold nanoparticles. So we were basically speculating and we were developing different kind of a hypothesis, and we were testing those hypotheses and so on here. So, based upon that, we just talked about that, that arsenic gets inside the plant cell using phosphate transporter. So we thought that, is some kind of one of the essential nutrient transporter being used by gold salt to get inside the tissue? On the basis of this, toxic arsenic mimics the phosphate and use the phosphate transporter to get inside from that reading here. So using kind of taking a piggyback ride here, that's what it is. And this was done by Dr. Ajay Chen. Currently he's working as a deputy director at MIT University in Jaipur. So we decided that why not we go ahead and look for the iron transporter. Okay? So we look for the iron transporter. There are few iron transporters being involved. IRTs is used for iron transport. There are some zinc transport that, we call them zip genes. They are involved in transporting zips for the zinc and IRTs for the iron. There are some other transporters which we call them FRO2 and so on, involving the iron transporter. So look here, develop a method by which we took these Arabidopsis particles and you grow them in control, grow them in AU10 ppm and cadmium 10 ppm. Reason we use cadmium is because just as a positive control. So our experiment is working. Okay? We harvested the root and we did the real-time PCR, looking for those different kind of transporter expression. And in this case, if you look at here, this is the plant cell. If you look, this is the plant cell, root plant cell. That FRO2, converts the iron and copper into single, into cuprous and ferrous ion molecule converted, and then these are being brought inside the cell by IRT's gene, IRT's transporter. Sorry. Okay, so we try to start looking for the expression of FRO2 gene, and if you look at here, this is the control here, FR2, and then when we take that 10 ppm tissue, 10 ppm gold salt tissue, that there is an expression of this gene here. So somehow FRO2 is getting induced in the process. Then we try to, if you look this one again here, FR2 is converting them from ferric to ferrous. We think, the gold 3 plus being converted to gold 1, and then it is being brought inside by IRT1 and IRT2 genes inside to convert them into gold nanoparticles. So basically same experiment was done, and if you look at here, control AU10, cadmium 10, and basically we studied the gene expression for IRT1 and T2, and if you look at here that these IRT1 and T2 are expressed in gold 10, but not expressed. In this case, CD10 is expressed, and CD10 is expressed in these scans. In this case here. So what we see that somehow in presence of gold salt within the medium, that certain iron genes are expressed. So we hope from there, it's not a direct evidence, but from there we thought that somehow the iron genes are expressed to bring these gold salt inside the tissue. Okay. We also looked for the other metallothionein genes. That whenever the plant is exposed to certain metals, certain metallothionein genes are expressed to protect the plants here. And we try to look for that here, and see in case of all these metallothionein, nothing happens in case of gold. But metallothionein, it is very well known that when the plant is exposed to the cadmium, metallothionein genes are going to be expressed to protect the plant, because metallothionein proteins will bind to the cadmium to protect the plant. So it's nothing is happening in presence of gold. Okay, so that was one experiment. Another thing is that we started looking transcriptomic study. We started looking that RNA is study, that what kind of RNA is being expressed. You can find it. Okay, and we also started looking the proteomics study, what kind of proteins are expressed in presence of the gold salt to the plant here. Okay, and we did that thing into different time, 12 hours, 7 days. These are the different upregulated gene, downregulated gene. We know the this is called short term, this is called long term, and then these are some of the gene expression, and from there, from this study as well as the proteomics, we came up that that certain genes are expressed in presence of gold: glutathione S transferase, thioredoxin, superdioxide dismutase, glyoxylase, oxygen responsive proteins, SOD, so on. So we basically took the transcriptomic data and the proteomic data and came up, tried to match those two together. Because, remember that you can look for the transcriptomic data, you can see the certain mRNA high, but that mRNA may not make a protein. Okay, so that's the reason we decided to do both of those, and from there that mRNA is present, at the same time proteins are present as well from there. Okay, so these are the some of the key proteins. You can see that that all these glutathione S transferase and so on. Okay, at the same time I had Devesh Shukla. He was another postdoc. He was working on different projects, but he was very good with the bioinformatic analysis, very good with that. And he took the data and he did the bioinformatic analysis, and he found that ABA responsive cis-regulatory element, we call them ABRE, is identified upstream region of genes in regulating gold salt treatment here. So it looks like the abscisic acid is also involved in this process. And then he came up with a model, he developed a model that from how gold and ABA is involved and taking the gold inside, became more kind of academic data, he started looking for the pathway and so on. Okay. So we we knew that, that what is the pathway for gold salt to get inside in order to be converted to the gold ion. But we still had the problem that extracting these gold nanoparticles from the tissue. And that's where we generated the gold, so we challenged your extraction of gold nanoparticle. And we needed to find out, and we just talked about that, we came up with the idea using a plant cell culture method. Plant cell culture method, easy to maintain, less interference to external environmental. You can do it in corner of labs, the small place. You can have a medium, and you can set up the whole experiment over there, okay? And then you can basically making sure that nothing contaminates your culture or tissue, okay? So we use these cell culture, and you can look at here the 10 nanometer, 50 nanometer, 100 nanometer, 200 nanometer. You can see the different sizes, shape, and so on. We also started looking that, average diameter in nanometer and concentration. So, that if you want to make a large size nanoparticle, we use the higher concentration. Look here, higher concentration. You can get up to 60, 70, okay? If you want the smaller size, use one of the lower concentration. We also started looking that, where is the toxicity. Remember that, one of the risk management, that we needed to find out that where is the toxicity. And the toxicity we were able to find somewhere between, anywhere between 10 and higher days. Okay? There was no toxicity before that, you can see here. So we develop these cell culture, we let them grow, then we break those cell lines, okay, and then using this column chromatography, you see that, and I brought this picture for you guys, column chromatography, we were able to load it here at the Cephadex, and then we were able to wash it, and it was fairly clean, having no biometrics. So we were able to do it. A lot of people, and this work has not been published yet. It should be coming out soon. So nobody knows about this thing, because many people were not able to extract them. They decided to leave this area, and we were kind of persistent and still working on that. Okay, so what we did, now we develop a method by which we can extract these fairly pure form, and then now we have pure stuff, you can do whatever kind of analysis you want to do it. Okay, so basically if you look at here, and I'm not sure whether you guys, this is a mice here. Can you guys see that from the back? Okay, so mice, we wanted to take these nanoparticles, and we started injecting into the mice. Okay? So far we had no idea that, because these nanoparticles, they were synthesized by the plant cells. So when they are synthesized, that, that when you extract them, what kind of coating does it have compared to the one you buy from a vendor by chemical method. Okay? Uh-huh.
When you are purifying using the gel chromatography, so you will be getting by size wise.
Yes. Uh-huh. Uh-huh.
So what is the type of triangular, what is the size of the triangular?
It is about 35. Remember that earlier I showed you?
Okay.
About 35. So it's not fairly pure yet. in terms of the triangle shape.
Right. But
Yeah.
we have different size of triangular nanoparticles, right?
Oh, okay.
So in that case, how it is purified?
Okay, so we have not gone that far yet, because our goal was to develop a method by which we can get the purity stuff. And now we have developed a method so we can go further and try to make it whatever 80, 90%. We still need to work on that manipulation.
So the same 35 nanometer is
In here, yeah, in here. So we took it, and again here that, being a plant scientist, I don't have any idea how to inject these nanoparticle to the mice. So I took to a guy who was animal scientist, and he helped us with that. So we injected mice, and then we started. So if you remember that, whenever the foreign object get into any living system, what happened? Anyone? All kind of inflammation response occur. Different kind of cytokine is going to be synthesized. Okay, so that's what we did here. If you look at here, that these are the gold nanoparticle in animal system, and if you look at here, these are the different kind of cytokine. All these over here, these are the measurement over here, and look here that the green one is plant synthesized, the red one is chemically synthesized, and the control over here. So if you look at here, all these are low, approximately to the control, or in some cases lower than control. So somehow when you inject the plant mediated synthesis of nanoparticle to the mice system, it is being recognized, and we think that because you see less response, less inflammation, less cytokine expression, it means we think that the living system thinks that, hey, you are part of us. You are not a foreign object. Because when you synthesize, when you put these, the chemically synthesized one, when you get inside, the animal system thinks that you are a foreign object. As a result, all these inflammatory response will occur. But if you look at in case of these, the green one, it is as equal to the control, or in some cases lower than the control. So what is happening? It's the same gold nanoparticle. What happened here? We have no idea. We think that that when they were synthesized, during synthesis, the gold nanoparticle were made, and then there was a coating around it, and those coating were still intact when we separated them through the column chromatography. So if you remember that, that you might have heard about that, these gold nanoparticle, when they get into the living system, you hear all kind of toxicity report. But in this case, it is not toxic, plant mediated. Okay. We also tested in animal cell culture. If you look at here, that these are the. If you look at here, these are the cytotoxicity on the normal mammalian cell. If you look at here, these are the black one. The black line is 24 hours. The red line is 48 hours. Okay. These are the cell. Normal mammalian cell, you see very little toxicity. Okay, this is the commercial one. So see that at 25 milligram per mil, that the maximum we have at 24 hours is approximately around 50% over here. In this case, using the commercial one, the one you buy from the vendor, in this case, it is around 35% down here. Okay. If you go to the cancer line here, see that this is your plant mediated, and this is commercially used nanoparticles. And if you look at the plant mediator, it is a lot more toxic to the cancer cells here. Why is that? I have no idea. I don't know. Okay? This is a lot less toxic. Okay, so this is basically how we did the experiment. I just put it here for you guys to see it. Okay? Another thing is that if you remember, and I guess earlier we were talking about, that if you are going to use these nanoparticles for any kind of potential role in biomedicine, okay? And if you are going to tag certain drug to these nanoparticles, so you need to know what kind of coating is there, right? Let's say, is it protein coating? All these functional groups are sticking outside where you can tag in whatever the drug is there. So in order to know that what role this nanoparticle will have, we need to find out what kind of coating is there and what kind of functional groups are still available to attach any kind of conjugated medicine or pharmacy, okay? And that's where, again, remember that I'm a plant scientist, I'm not a chemist, so that's where I'm stuck right now. We tried to do whatever we could, and my postdoc, he basically did it, this FTIRA scanning of surface functional group, and from there he can find out these are some of the, these are some of the functional groups that, he can find out. Presence of aliphatic alcohol, alkyl, hydroxyl amino groups present here. But that's all we can say that, and I'm hoping that, that to the new place that, I'm going to be, this has a great pharmacy school, so hoping that, they should be able to help me analyzing that, what is the surface coating on that nanoparticle. Because remember that there is something, that's the reason it is showing less toxicity or no toxicity compared to the same nanoparticle coming from the chemically synthesized method. So we need to know in order to have any kind of functional, any kind of potential role in the future.
Did you do the cleaner spectrum for the chemically synthesized nanoparticles?
No, we have not. But we need to do that. Yeah. But for chemically, that when you buy, they will tell you that hey, this is the particle and these are the coat are stabilized. Yeah, you can find it.
They will also give the FTR spectra.
Yeah, so you can find out. You don't need to do it, because generally.
Sometimes we can find some information. That's what I asked.
Oh, okay. Okay. So just to conclude, this is the story I wanted to tell you guys that that's where we are right now. I'm still looking for several collaboration, several interaction with the different people, having expertise in different area to answer certain question for us. Okay. So just to conclude that yes, plants can do synthesize these nanoparticles. They are green method, they are sustainable method. Different genes are expressed differently to control the shape and size of those gold salts. Different transporters are involved as they move, these salts move from outside to inside. Characterization of particle essential. We need to know that that when we synthesize, what kind of coating is there on those nanoparticles. And again, just being an expert in one area that you cannot be successful. You cannot be. I mean, that's what I'm stuck. And I'm trying to find out from different people that how they can tell me. Okay. But again, we need to do, I mean, it looks promising. You see that we injected in mice, and it shows less inflammation compared to the chemically synthesized one. So it's a relevant, less toxicity, but we need to know more about that. We need to do more study about that, that, yeah, it is good for the mice system, but do we know that can we take it to any other animal system? Is it going to be good for the human system? We don't have any idea. Okay, so there is still a lot of knowledge we need to gather from different experiments about this. Okay, and again, as I said, my God, it's interdisciplinary. I mean, this guy here, John Andersland, he's the best TEM SEM guy. He did his PhD from Cornell University. Okay, very helpful to work with the student. Okay, so he helps. Jorge Gardia, analytical chemist. There was no way for me to find it out that these, we see the gold particle and those gold were AU0. He's the one who helped us. Paul from IIT, Kharagpur, that yes, that they are there and they have catalytic activity. So there is some potential for that. So, and then over time I have also collaborated with different people. I'm still looking for collaboration with other people which can help us. And then nothing is possible without the funding, right? Funding, you got to have the funding. And I'm really appreciate you for National Science Foundation Molecular and Cellular Biology section that they gave us. I had one finished 3 years. Now I'm the second one. Then hopefully I may need it for future. And then my older university, I moved to another university. Older university was very helpful in, giving some money to help me with the these programs. And I used to be assistant director of applied research and technology program, and through that program we used to buy different kind of instrumentation for the biotechnology research in the campus, and I use many of those instruments to run my experiment. Thank you. Any question?